Hyung Gab Cho from Ina University in Korea. Today, I would like to about three species classification using hyperspectral and multispectral imaging. Firstly, the hyperspectral data compose the number of band and neural bandwidth that is useful to discriminate forests that possess biochemical and biophysical characteristics. The hyperspectral data than more than multispectral is oversampled. For explain completeness characteristics of targets. And multispectral data is undersampled because of broadband and non-continuous spectral band. If you saw, what data do we need to use? In our previous studies, one study conducts classification using two hyperspectral imaging and obtained better results than using single imaging. And through comparison with other data, explain higher accuracy of hyperspectral data. But the previous studies couldn't prove as high accuracy quantitative, but also comparison data acquired at different time. Therefore, we are required new study for quantitative analysis and at the same time. In this study, we are trying to investigate comparability between hyperspectral image data and multispectral image data for species classification. Major material of this study is KNA, the Korean National Arboretum. This area is located in Gwangneung Experimental Forest of the Korea Forest Research Institute in South Korea. Among them, we acquired this image that compose mainly deciduous species. Aforementioned image is Kashi 1500 urban hyperspectral data. It taken in October last year. It consists of visible and near-infrared wavelengths range of 19.6 of spectral band and 1.5 meters spatial resolution, and have 40 degrees field of view. Also, for acquired multispectral imaging of same time and same area and higher spatial resolution, we tried simulation about Landsat-TM using Kashi data. We convert Kashi corresponding to Landsat-TM band 1 to band 4. For simulation of Kashi, we use the spectral response function. This graph shows spectral response function of each TM band. It could be found that each band presents Gaussian distribution. After acquired spectral response concerning TM band, to conduct simulations through this equation. As a result, figure shows simulated TM band 1 to band 4 imaging. Band 1 consists of blue band of 450 to 520 nanometer, and band 2 is green band of 520 to 600. Similarly, band 3 and band 4 is composed red and near infrared bands. For comparison of above simulated TM and Kashi imaging, we try collection training sample. We collect 12 deciduous species with characteristics of homogeneous and free-form condition. Each hard would stand as a size of sub attached to a few target area. Each species is collected 10 training and 30 test sample. As you can see, sample is considered to collect sunny side of canopy. Also use reference data, Gwangneung forest map, and area photo, and field survey data. For the study, we use the same classifier of various classification methods. Same, the spectral angle mapper, compare with spectral vector corresponding target of individual range. Sam assumed these species have unique spectral identity of spectral signature and difference of spectral signature of species. If spectral angle is smaller than tolerance value, to cite target is massive. Same classifier is useful when using hyperspectral data because of not consider number of training samples. Maximum angle of same parameter represent to a low spectral angle. In this study, maximum angle is 0.1. Results from classify using CASI and simulated TF. Show classify our area as shadow area, and several conifer species. But the target area comparatively classify well. Show pattern only for 12 deciduous species. This figure shows masked area except for 12 target classes. CASI result better classified and simulated TM result entirely. Especially of two classes, a Jacoba forest stand of blue color and Lura of purple color. Figure shows CASI is well classified and simulated TM. Following quantitative analysis, Passer's classification approach. Table shows producer approach for each 12 species in 4 species. CASI classification result shows over 10% higher accuracy than simulated TM. And CASI classification results show accuracy of over 50% in all of deciduous species. Also, in comparison of overall accuracy acquired using CASI higher than simulated TM as difference of about 9%. But as mentioned earlier, difference of 9% of classification accuracy couldn't explain the CASI result is better classified than simulated TM. Then, is this difference statistically significant? For verify statistical significance of classification result in 1983, we tried to use G-test proposed Gongdae-Ton. He proposed that conduct G-test using individual kappa coefficient and variance of confusion matrix. When level of confidence of 95%, if observed G-value is bigger than 1.96, difference of two classification accuracy would be reliable. This G-test apply previous obtained classification result. G-value is calculated over 12 statistic value. Therefore, at the confidence level of 95%, difference of classification accuracy is significant. And CASI could show better classification results than simulated TM2. Yes. Finally, conclusion of this study is that classification accuracy of Kashi image was significantly higher than similar TM image. And high force factor image has more capability for 3 species discrimination than multi-spectral image that could contribute to generate detailed forest map. In future study, we will conduct compare with other types sensor data or other more than species or with increased sample. Thank you for your attention.
Thank you. Thank you for your nice presentation, Mr. Chao. Okay, this presentation is open for this discussion. So, is there any comment? Okay, yes, please. Yes, please.
Question: could you please show your entire study area? How many tree species are there in your study area? Why didn't you include other tree species and the 12 tree species in your classification? Thank you.
In study area, this area consists of 27 species. Deciduous species and conifer species.
Let me explain as a cause of this paper. And this is the experimental forest, and the site is about 20 deciduous species. All other stands are natural forest. So that we just exclude the natural stand, and then include only 12 deciduous species, which is not usually capable to classify with any other data set. So that to exclude some complexity on natural stand, which is not quite met. But this is a plantation stand, which is developed in Korean Forest Research Organization for a long time ago.
I think it's more difficult to classify natural species.
Of course, it is from previous study, you said it's on the natural stand. But the problem of classifying natural stand is we can classify, but do the evaluate classification accuracy and those kind of stuff are sometimes very time consuming and very difficult.
Well, you did perform that in Taiwan. I think it's very, very useful. Thank you.
Okay, yes, please.
Thank you for a nice presentation. Could you tell, I have 2 questions. First one is about, could you tell me about the size of the training sample? Size of this training sample. What was the size of training sample?
It's
No.
point.
Training sample 몇 개 썼는데 테스트 sample 몇 개 썼는데. Next. Okay.
10 training sample.
No, I am asking size. You just mentioned 10 training sample, not the size. It was 1 into 1 meter and 10 meter, 30 meter.
No, that is a 10 sample pixels for each species.
So the pixel size was
Pixel size is 1.25 meters, 125 centimeters.
like in case of deciduous forest, like most of the forest having not very dense canopy. So did you consider any reflectance from the soil?
Well, this is a very dense canopy. It's a very well-grown trees.
But
So
still, like in case of deciduous, they are not very dense as compared to evergreen, and always the most of the reflectance comes from the soil.
No, not so. This is fully in terms of closed canopy situation. previous papers using some different classifier and some different types of hyperspectral data, which is maximum likelihood classification in previous paper. But this paper we used the spectral angle method. But interestingly, we can see very similar trend between two studies, although the study area is quite different.
Okay. Yes, please. This is the last one.
I just have one comment. You compute many statistic feature, but you use 4 digits after decimal point. It is meaningless. I think just only 1 or 2 digits enough.
I don't quite understand it.
The computation, you use very 4 digits after decimal point. You show some accuracy or some. This is
You mentioned the accuracy.
next, maybe some other. This is about overall accuracy. This is meaningless. This is 4 digits after decimal point. Okay, so maybe at the plus
Okay,
point.
let's say just a 75, 67. Okay?
Okay. Is there any question or comments? Okay. No. Okay. Thank you, Mr. Cho. Thank you again. Okay. We move to next presentation.